So our next speaker is Navin Kapur from Tufts Medical Center. Navin is an interventional cardiologist who has focused a lot on cardiac fibrosis over the years and now is looking at unloading as a way to modulate molecular mechanisms in the setting of acute MI. Navin?
Great, thanks very much. So the topic that I'll be talking about for the next 15 minutes is primary LV unloading and the mechanical conditioning hypothesis, which is the term that we're using in our laboratory right now. So as you've heard about this morning, there seems to be a paradigm shift that we're all talking about in terms of interventional therapies for acute myocardial infarction. And part of this is that part of the reason for this is that over the past decade, what we've seen is that there's been a transition in our clinical outcomes for patients who come in with acute MI. What we're seeing here, and this is highlighted by a study by Attenuates in 2009, looking at 8,000 patients coming in with acute MI who were treated with early PCI therapy. And what we're seeing is that there's a significant reduction in the amount of in-hospital and short-term mortality associated with MI for patients who get treated with PCI. But what we're also now learning is that we're creating an increase in the burden of ischemic heart failure. And this is both in the acute setting. In-hospital heart failure has increased over the past 2 decades after MI, as well as heart failure within the next 5 years. So this burden of ischemic heart failure is now, as Dr. Brunwald said, is the new enemy for myocardial infarction. And as we've talked about also today, you're hearing a similar theme for this first session about the current paradigm focusing on rapid coronary reperfusion to limit myocardial damage. And of course, this is focusing on half of the seesaw, which is oxygen supply, and the target that we're looking at for unloading is actually oxygen demand. What's also important to recognize is that more aggressive door-to-balloon time reduction doesn't seem to be the way to go. So there's been a significant push towards getting door-to-balloon times well below 90 minutes. I think most centers are now achieving that number. But what we see is that there's no impact on mortality with the reduction in door-to-balloon times far greater than reduction less than 90 minutes. And of course, also taking from Dr. Brunwald and Dr. Kloner, this term, the double-edged sword of reperfusion, is what we've really started to focus on in terms of our work with mechanical unloading. And so I think the fundamental principle here is that, which we all agree upon, is that time is muscle. And the reason why we say that time is muscle is that when an acute coronary artery becomes occluded, that ischemic insult starts to activate a mechanism within cardiac myocytes whereby there's a decrease in oxidative phosphorylation, a loss of ATP synthesis, and this leads to a decrease in intracellular pH due to calcium influx, lactate elevations within these myocytes. And as a result, there's downstream effects on mitochondrial function where the mitochondria essentially become dysfunctional and can actually burst in many of these scenarios. And this results in cell death and necrosis. And the treatment right now for stopping this myocardial injury clock is to reperfuse. But it was asked earlier by Dr. O'Neill about whether or not reperfusion actually exists. And what we're saying is that when we reperfuse ischemic myocardium, we're actually creating a feed-forward mechanism whereby there's worse mitochondrial damage in cells that are susceptible, and there's also an increase in oxidative damage as well. Now, the body has an inherent counter-regulatory mechanism. The first is at the endothelial level, where endogenous TPA tries to autolyse thrombotic occlusions. But also at the cardiac myocyte level, there's a significant increase in salvage kinase pathway activation. And these kinases are listed in the right side where you can see AKT, ERK, glycogen synthase kinase 3 beta, leading to an anti-apoptotic signaling pathway. And this is designed to counteract the effect of reperfusion injury. So as we said, right now we're stopping the myocardial injury clock by reperfusing the artery as quickly as possible. And the question also was asked earlier, as we've focused on hemodynamic effects of these pumps, but now the question is, are these pumps actually affecting biology? And are they affecting some of the pathways that have been well established in terms of preserving myocardial salvage and reperfusion injury salvage pathways? So that's what our laboratory has been focused on, and a lot of laboratories around the world have focused on this. So Noam and Jerry put together this slide which basically summarizes the number of drug trials trying to attempt cardioprotection in the setting of AMI, and what they found was that there were a lot of trials that essentially have not worked. And critical barriers for these cardioprotective approaches using drug therapy is first the mandate for rapid coronary reperfusion. So by the time you get the drug on board, does it actually get to the target? The second is the inability to target multiple cascades that are affecting reperfusion injuries. So if you take one kinase and you give a drug for that one kinase, is that really sufficient to give you enough protection? And then, of course, managing hemodynamic instability. These are patients who are having an ST elevation MI. The hemodynamic aspect is not dealt with using cardioprotective drug therapies. And this editorial, a review article that just came out in August, there's another accompanying one in circulation as well, asks basically the question we're all asking here. Is it time to give up on cardioprotection? And I hope that our resounding answer after today is that it's not time to give up. But this is a critical appraisal of mechanical approaches such as preconditioning, post-conditioning, and remote ischemic conditioning, and we'll be hearing more about this later on. But my takeaway when I read these review articles is that we have to come up with new approaches for cardioprotection in AMI, especially given the increasing burden of ischemic heart failure. So when we think about novel paradigms, again, this is a theme that you've heard through the talks this morning. There's a clinical objectives that have to be met with acute MI. One is taking care of the acute coronary occlusion, and the second objective is to manage hemodynamics and hemodynamic stability in the patient. This, of course, is now balanced now as we're introducing this concept, and I really like the term functional reperfusion as a way of targeting myocardial oxygen demand from Kenji, where we now are not just focusing on restoring oxygen supply, but now we're trying to see, can we modulate demand in this setting? So we know from the surgical approach in terms of ST elevation MI back about 30 years ago, there was actually very nice data coming out of the operating rooms that if you first take a patient who's having an ST elevation MI to the operating room, they actually do quite well. And in the surgical management, it's first unload the patient, you put them on cardiopulmonary bypass, you then put basically bypass graft, and that takes time. So there's a delay till you actually get to the reperfusion phase. But ultimately, you do restore coronary perfusion. So this surgical approach to revascularization in MI, of course, was lost once angioplasty came on the scene, and the focus again became driven towards restoring oxygen supply. But first unload and then reperfuse is actually a surgical mantra for the management of acute myocardial infarction. So when we bring that surgical mindset to the interventional community, there was some early work done in the late 1970s, early 1980s. This is some work by John Laschinger, who's a surgeon, who actually was doing some of the earliest work looking at left atrial to femoral artery bypass as a mechanism of unloading. And what he found with these surgical models in canine models was that by initiating left atrial to femoral artery bypass, that they were actually able to significantly reduce the infarct size compared to the area at risk. And this was one of the first studies that actually highlighted this fact of mechanical unloading as a therapeutic approach in the setting of MI. And of course, over the past decade, what we've seen is an absolute significant increase in the number of percutaneously delivered circulatory support devices. And these are now, the ones that we're focused on are the continuous flow pumps, which are illustrated here. So in our laboratory, we wanted to look at what has been the history of mechanical unloading in acute MI. And this is actually not a new concept. The earliest work was done by folks like Richard Smalling, who started off with work with the intra-aortic balloon pump and the hemo pump in the 1980s. And what Dr. Smalling in his laboratory showed was that if you initiate mechanical support during ischemia and reperfusion, that of course you can reduce infarct size. And that was one of the earliest observations of device unloading therapy to reduce myocardial damage. The limitation of this model, of course, is that you're activating these devices at the onset of ischemia and keeping it on throughout the period of reperfusion, which translationally makes it a bit challenging. But further studies have been done. This was by Bart Mines in the early 2000s in a surgical sheep model. And now the importance of this study was actually that they used the axial flow pump as an LV direct unloading mechanism. But what they focused on was not only the activation timing, so should it be done during reperfusion alone, but they also looked at this concept of total and partial unloading, which you heard about from Kenji and Dan this morning. And the study essentially showed that if you can get significant unloading, and this back then significant unloading was 4 liters per minute, that you would actually see a substantial reduction in infarct size if the device is activated during reperfusion alone. And that's a much more translatable concept than activating it at the onset of ischemia. So this was one of the first studies that started to translate the science of mechanical unloading as a mechanism for infarct size reduction into the clinical arena. So we started looking at this data, and we've been working clinically quite a bit with a number of pumps. We started off with the tandem heart device at Tufts Medical Center, and we found actually that the tandem heart device worked quite well in terms of mechanical unloading. This transeptal approach with left atrial unloading was key. And our initial hypothesis was that if you can initially reduce LV work and delay coronary reperfusion, that you would limit myocardial injury and acute MI. And I put an asterisk next to that concept of delay because it actually turns out that the only reason we delayed reperfusion in this setting was that we wanted to replicate what would happen in the cath lab if you were to put a tandem heart device into a patient. And as Dr. Brunwald illustrated, this requires a transeptal puncture. It's 2 large bore cannulas, one going into the left atrium, one going into the arterial system. And it takes time to stabilize those cannulas after you activate the device. So we knew that in order to translate this preclinical study into a clinical trial, for example, we would have to impose a delay to coronary reperfusion. And it turns out that that was a fortuitous thing, because what we found was that this delayed reperfusion, although it was being driven by necessity, actually may be one of the critical components to translating acute unloading in the setting of acute MI. And so this was the animal model that we had set up. Unlike other models that had been published at the time, this was a closed chest model, everything delivered percutaneously, so to mimic the cath lab as much as possible and reduce the inflammatory damage from open chest surgery in these models. And what we found was a significant reduction in infarct size that correlated with a reduction in LV stroke work. And for me, that was the first insight into the fact that we can modulate demand and actually have that impact myocardial infarct size. So Bob Kloner wrote the editorial to go along with this, this paper, and as is often the case, these editorials are very insightful. And what he asked for, first of all, was that these were small n numbers. Is it clinically feasible to do transeptal LAFA bypass in the setting of an ST elevation MI? What's the optimal timing of delayed reperfusion, and what's the mechanism? We had limited mechanistic data in this paper, and specifically he wanted to know what's up with signaling in the infarct zone. And so I took these questions from the editorial and I said, okay, well, this is our next set of studies that we're going to execute. And so one of the things around the same time we had been studying the effect of left atrial unloading versus LV unloading and comparing the Impella to the tandem to ECMO. And with all of that insight into the mechanism using hemodynamics, what we found was that the Impella CP device gave us a very effective unloading signature. And you saw that with simulations from Dan. We see this clinically, and we also saw this preclinically, where we saw a reduction in LV pressure and LV volume. And so that reduction in PVA, we could now achieve without having to do a transeptal puncture, and we could do it with a single stick arterial access that was much more feasible. So we then said, let's design a study where we're going to test the limits of primary unloading. And a little bit to the dismay of Abiomed, I really put the burden on the Impella device with this experimental design. Because what I said was, we're going to put the Impella pump in, but we're going to extend the time of LAD occlusion to 150 minutes. And we're going to compare it to a control arm, which only had 90 minutes of LAD occlusion. So the burden of proof fell on the Impella device to demonstrate whether or not unloading and delayed reperfusion actually reduced infarct size. And what we found was that when we looked, first of all, at infarct size reduction, there's a significant reduction in the primary unloading group compared to primary reperfusion. We again saw that there was a signature that there was a correlation between infarct size reduction and LV wall stress in this case. But what we also learned is that there's a significant amount of biology that's occurring that gets activated when we activate the Impella pump. And specifically what we found was that myocardial levels of a chemokine known as stromal-derived factor 1 alpha, or SDF1 alpha, were increased in the animals that had the pump activated, and that the regression plot of infarct size to SDF1 levels in the myocardium actually was almost linear. And so that was one of the first insights that we're actually seeing a biologic effect of mechanical unloading in the setting of acute MI. And so that took it out of just pure hemodynamics and started to introduce a new direction for this field of cardioprotection. So with that work, we came up with this concept of the mechanical conditioning hypothesis, where first unloading the LV, delaying reperfusion, activates a cardioprotective program that limits myocardial damage in AMI. And the biology is shown here, but essentially what we saw was a significant reduction in myocardial damage driven by some of the same pathways that are associated with canonical cardioprotective signaling through the risk pathway. So this, of course, led to new questions. And so the next question was, well, how important is this delayed reperfusion? Do you really need to do that? Because designing a trial where I asked Bill O'Neill to stand there for 1 hour and not open an artery did not seem like it would be something that I'd be able to ask most interventionalists to do. The second question is, how important are the kinases? Is this more of a hemodynamic effect, or is this actually a biologic effect that we're seeing with these pumps? And the final question is, what's the long-term impact of LV recovery? So we showed acute phase myocardial infarct size reduction, but what happens a month later, for example? So that's the work we're doing now. And the first question, looking at this timing interval of whether or not you need the delay, we studied this by essentially looking at a dose-response curve, where we took a series of animals, the same swine model. We had a group with ischemia-reperfusion alone. We had a delay of reperfusion after activating the Impella device by 15 minutes, 30 minutes, and 60 minutes. And then in the final group, we activated the Impella device after balloon reperfusion. And so the idea was to see what actually do we need to do in terms of this timing interval. And what we found was that you need to have about 15 to 30 minutes of primary unloading to actually see the beneficial effect of infarct size reduction. And that told me that something's happening in those 15 to 30 minutes that's allowing for this activation of this cardioprotective mechanism. You can't just put the pump in after reperfusion, and you can't put it in right before reperfusion. So we looked at the biology, and this is unpublished data that will be coming out. But essentially what we found was that the same sequence of that mechanical conditioning pathway, SDF1, its receptor CXCR4, and downstream signaling through AKT, GSK3 beta, were all activated if you put the pump in before reperfusion and if you delay by 15 to 30 minutes. So this next question was, how important are these kinases? So in this set of studies, we actually went in with a over-the-wire balloon system, occluded the artery, and then through the over-the-wire system delivered inhibitors of specific kinases in the risk pathway. And what we found was that in these 4 bars, all of the animals had unloading for 30 minutes before reperfusion. And in the second bar, you're seeing this inhibition of AKT and ERK kinase. And in the third bar, you're seeing inhibition of the SDF receptor CXCR4. And what we found was that if you take away the activity of the kinases, or that signaling pathway, you lose the beneficial effect of the infarct reduction with the pump, suggesting that there is a biologic component of this hemodynamic pump that's actually impacting infarct size. And in the final group, we tried to deliver SDF1 through the coronary to see if we could augment the reperfusion effect using a gain-of-function approach, and found that we could not get the infarct size to reduce even further by giving more SDF1 in this setting. So Dr. Esposito later on will be showing our next analysis, which is looking at the microarray transcriptome that we did with these series of studies. And what we found was that primary unloading actually triggers a global gene shift in expression within the infarct zone itself. And so you'll be hearing a little bit more about that later on. The final question was, what about acute unloading and its impact on LV recovery? So what happens 30 days later? And so we designed this study where we essentially replicated our Impella to balloon model with 30 minutes delay, waited 28 days, and now we're looking at this data for MRI and a number of other metrics for infarct size reduction. And this is some of the preliminary data. What we're seeing here on the left is the primary reperfusion group. That's the scar 28 days later after our model of LAD occlusion and reperfusion. And in the primary unloading group, there's a significant reduction that's illustrated in these images in that scar size 28 days later, similar to what Kenji had shown histologically or pathologically with his neuromodulation approach. When we look at function, what we're seeing is that on the primary reperfusion group, there's that classic anteroapical hinge point due to an LAD infarct pattern. And that also now, of course, compared to the primary unloading group, where we're actually seeing intact myocardium. You do still see a little bit of that smaller scar hinge point there, suggesting that we are actually potentially able to impact LV recovery with this model. So this is some of the preliminary data. This is only an N of 3 per group, so you won't see any statistics here. But the first panel there shows this reduction in late Gatton enhancement measurement of LV scar. And this is the raw data. We have these blinded in terms of the readers of the MRIs. What we're seeing is a significant reduction in LV scar 28 days later after primary unloading compared to primary reperfusion. So when we think about this concept of going from door to balloon to door to unload, focusing on myocardial oxygen demand, functional reperfusion, as opposed to direct coronary reperfusion, we still believe this concept that time is muscle. But what we think actually now is that primary unloading introduces an element where you can stop that injury clock, at least pause it, and potentially activate a protective pathway that will limit the damage once you reperfuse the artery. And so that's what we're going to be working on for the next few years, and we're hoping to translate that into a clinical trial. A number of the focus of the ACURE group is to develop that trial, and we have some talks about that later on this afternoon. So in conclusion, the burden of ischemic heart failure will grow. New approaches to cardioprotection and AMI are needed. Primary unloading reduces infarct size through a 2-pronged approach. First, reducing the wavefront of myocardial ischemia, and second, by activating a cardioprotective program. And prospective randomized control studies are required to test the validity of these preclinical observations. So thanks very much for your attention.
Thank you, Navin. Sal?
Yeah, Navin, tremendous talk as usual. I had a case about a year ago of a 35-year-old gentleman at one of our satellite hospitals. Drove out there, it was about a 25-minute drive, big anterior infarct, cardiogenic shock, you know, typical scenario. Shot his coronaries, flush occlusion LAD, EF of 20, EDP of 40. I put in a CP35 up front, open LAD, the patient does well. Okay, essentially during the case, total uncoupling, full Impella dependence. I get a call from my program director the next day saying, your daughter balloon time was 92 minutes, okay? And I got raked over the coals. And it's, you know, it's become this issue where one of our biggest nemesis is that daughter balloon time. And I was under the assumption that it was excluded from NCDR, which it's not. We went back and, you know, they've surveyed that. And, you know, in our area, actually, people are competing for daughter balloon times. It's advertisement. You know, there's people staying in house. Our daughter balloon times are 40 minutes. Come to us because we're better. Is there anything from this esteemed group that is being done to help with removing Impella from the daughter balloon time equation? And I think that's going to be huge in increasing this whole concept of unloading, because everyone's in a rush. And we get report cards on it if you look at our NCDR.
Yeah, so it's a great question, and it's obviously one that's very relevant clinically to translating the science. So I think the short answer is that the way to get over that limitation is a randomized controlled trial. That's a primary objective of the ACURE working group. We'd love feedback from everybody here today about the concept and what you think about it. I think right now, you know, when I read the STEMI guidelines, I think they're written appropriately. I think that this idea of mechanical unloading, even in the setting of cardiogenic shock, in the setting of ST elevation MI, is a new one. And that your scenario is exactly what we're trying to achieve by getting this data out there and putting together plenary scientists and clinicians to get the randomized controlled trial data out there. But short of that, I would say that the most important thing for you to do clinically on a daily basis is to document cardiogenic shock. And one of the things that we constantly forget about doing in the setting of mechanical support is putting in a PA catheter. And even getting a CVP RA sat that demonstrates that you have a low cardiac index is going to help. It won't completely solve the problem, but it'll help give you a lot more rationale for using mechanical unloading.
To do that in these sort of borderline cases.
Yeah.
We're not sure. We'll put a PA catheter up front, and if the mixed venous is 50, we'll say, you know what, this gentleman needs support. If it's 70, it's not. And again, that's led to some delays in daughter balloon time.
That's right.
And that's, again, that's part of this issue. So I would appreciate any help from the panel here.
We're here to help.
Andreas?
Navin, congratulations for this very nice project. Do you have an idea where the SDF1 comes from, which cell type?
Yeah, it's a great question. So, you know, this is when I submit this to MIMS, which is our myocardial ischemia metabolism group, this is the first question. And I try to remind them that an Impella doesn't fit in a mouse. It's actually very hard to do what we typically do in our molecular laboratories with transgenic models to get to this specific question. But I think that, you know, what we've found at least is that there's a substantial increase in the amount of myocardial SDF expression with unloading. We have looked at circulating SDF1, and the levels are elevated, but as you can imagine, it's diluted. And so we actually think right now our postulate is that this is coming from the myocardium. There are, of course, multiple potential sources for SDF1 in the circulation as well as in the myocardium and other tissues, which we have not yet looked at. But that's a question that we are focused on, and our science going forward will address that.
The reason I ask is that for recruitment of progenitor cells, we know that adherent platelets are the driving force for STF1 secretion, and if that would be the case, it would be very important to reproduce the results that you have seen in the presence of strong antiplatelet treatment, because that is what happens in the clinic when we insert the pumps as well.
Yeah, and there will be a talk actually here that I'm very much looking forward to on the use of thienopyridines in the setting of acute MI and infarct size reduction. So we'll hear more about the role of platelets. But specifically to SDF1 expression in platelets, we haven't measured it, but we are seeing an increase in the myocardium itself.
Eric, last question, then I'll make comments, and then we'll break.
Yeah, very nice talk. You gave this sort of early distinction between the failure of the reperfusion injury drugs and now sort of the success we're seeing with this unloading and delayed reperfusion. Is it an either or? Would it be the possibility of if you put in an Impella now, there might be a rebirth or opportunities for these drugs that we've tested in the past and failed?
Yeah, I'm smiling because, that's a great vision. It's very exciting. It's something that we've started testing in our laboratory, is what do you do during those 30 minutes? And do you just sit around? Do you, adjust the radio in the cath lab? What else could we be doing? And some of the earliest data, which I didn't show today, is we've actually now started combining early beta blockade with mechanical unloading. And what we're seeing is actually a much better effect in terms of this LV-AO uncoupling. Hemodynamically, it's beneficial. There's some phenomenal studies that have come out this year looking at esmolol, metoprolol, and resurgence just in beta blocker therapy, FDA approved, of course. But by having a hemodynamic pump in place, you can now give that drug, which we know is life-saving. And so I think that's some of the lowest hanging fruit in combination of drug and device therapy for cardioprotection.